عادة هي شيء شنخون بلان blood glucose level will remain in a narrow limit. Blood glucose level will remain in a narrow limit. If there is low blood glucose, this is called hypoglycemia. Hypoglycemia is danger, may lead to coma and death. There should be a normal blood glucose in the blood. كواتا blood glucose أبدا blood we have a range that maintain the level of glucose. High blood glucose is not good and low hyperglycemia is bad and hypoglycemia even worse. كواتا blood glucose أبدا level normal minute. How? What are the sources of glucose in case of fasting? Glycogen is a source of glucose for short-term fasting. There should be a bank يعني storage هبه كجلوكوز اللي هو كده. لشي هو كده glycogen. And glycogen is stored in which organ? Liver. There is a limited amount of glycogen, limited amount, in amount of milligrams. In amount of milligrams. كهذا كتوقعت برسيدة به blood glucose level تاب تدو ساعات يعني سيل شوار ساعات fasting بشي maintenance كده glycogen breakdown, which is called glycogenolysis, which produce glucose. How this glycogen is formed? Glycogen is a complex molecule of glycogen is a complex molecule that form from the glucose. These glucose are bound to each other by a glycosidic bond. We have two types. The glucose can pick one and say, Nagatera. Glucose attached to each other by glycosidic bond alpha 1,4 and alpha 1,6. Masama lechia number of the carbon rakami la sara carbon glycosidic bonds, we have 1,4 and 1,6. How? Look to this molecule. Amino glucosan attached to each other by glycosidic bond. Lebeni carbon number 1 and carbon number 4 of the others. Say the kema structure is our drusaka. Well, enzyme is a branching enzyme. Okay, boshash peoene. Lalakan carbon number 6 from the other side, carbon number 1. Kawata masama lachi glycogen is a complex molecule by several enzymes كدروسبه. يكيك لوانه بيوتي branching enzyme. هم branch عنه خاطر. Glycogen formation is important because it occupies small amount of space. المشكلة من مين؟ Glycogen is a complex molecule, should be packed in a certain space like liver. This is important to prevent osmotic effect of glucose. كواتا أجا glucose أجا هل أوبكو glycogen نبيه water إيش رأيك يا شيء لقى خويا كواتا glycogen بوشي دروس بوي osmotic effect كي كم تربي. This is the structure of glycogen molecules. Structure of glycogen molecules or granule. there is no specialized tissue for collection of glycogen. Glycogen stored in certain types of tissue like liver, kidney, very small amount in the kidney, and this can be used in case of fasting. The important point in glycogen, if there is any defect in the formation of glycogen or degradation of glycogen, there is a disease or metabolic disease or genetic called glycogen storage disease. If there is no breaking down of glycogen, what happens? There will be hypoglycemia because there is no glucose entering into the blood. Hypoglycemia in case of glycogen storage disease. لحد ستيبك إذا إنزيم كان deficiency هبه أجا branching enzyme إيشنكا جورة type يكي بيوتريا. Collection glycogen in the tissue cause what? In the liver cause swelling of the liver and damage of the liver and hypoglycemia. Man glycogen the quick dish the muscle shahaya inside the muscle is important. اگر glucose formation نبت يعني energy production نبت أبت هو muscle weakness and hypoglycemia ياريه كامنا وكا أكيد بعدها كما يتشبه due to hypoglycemia. Glycogen is a complex molecule of glucose. If there is any defect in the glycogen metabolism, degradation or formation, this is called glycogen storage disease. We have different types of glycogen storage disease. SAO بس أو نقطة بزن كديزيز يكيا بيوتي glycogen storage disease. هاتش enzyme deficient بيلا glycogen metabolism. بفي أو نابكي. We have different names. بس استجاريو ناو كان يلبر مكان صال دوائن تنميته وبدلها. Glycogen synthesis called glycogenesis. Glycogenesis, formation of glycogen. How this will form? First molecule of glucose is inactive. There should be activation of glucose. Gutman molecule can be activated. Phosphate glucose phosphate activation of an enzyme here phosphate Gutman phosphate kinase. This glucose 6-phosphate change to another structure. Gutman high conformation glucosakachi period. Isomerase. شو أنا كيف وضع يعني شو أنا كيف شو كبيرتي بقى يعني mutase glucose 6-phosphate change to glucose 1-phosphate and after that we have a structure which is high energy UTP change glucose 1-phosphate to UDP glucose. UDP glucose is a donor of glucose. يعني glucose كان high energy ده ما تيجي بيها أما serious reaction ده glucose كان إن بيكتري أبد هو يبيك glycogen تبص. We have two types of bond, alpha-1,4 glycosidic bond and alpha-1,6 also glycosidic. Awika branching naka. UDP glucose is considered as an active form of glucose, or donor of glucose. Look to this diagram, show you how glycogen is formed. Glucose 6-phosphate change to glucose 1-phosphate by mutase enzyme. Then UDP glucose will give glucose جارێک وا دروستە بێت جارێک برانچینگ ئینزایمێ 1,6. When glycogen is degraded, branching enzyme, just name of the branching enzyme. 1,4,1,6. Oh. With my glycogen is degraded in the skeletal muscles in response to what?
Exercise.
In response to exercise, when you exercise, you increase the glucose inside the muscle. And in case of fasting, امرو بالرجيتو لكاتي fasting پێویستە بە چی هەیە؟ پێویستە بە دیگرێدیشنی گلایکۆجین هەیە تاکو 3-4 ساعت پێویستی بە دیگرێدیشنی گلایکۆجین هەیە. ئەگەر دیگرێدیشنێ بۆ نەکرێبەوە ئەنزیمەکەەوە ئەبێتە هۆی چی؟ فاستینگ هایپۆگلایسیمیا. ئەو وتمان هایپۆگلایسیمیا is danger. Look to this diagram. Inside the liver, we have glycogen. Glycogen changed to glucose 6-phosphate. And finally, glucose will be released into the blood. While in the muscles, glycogen changed to glucose 6-phosphate to be used as energy, not releasing glucose. كواتر لمصلى جلوكوز ريليز نابي بونا وبلط بلان للليفر دروس الليفر أبيت هو يريد جلوكوز دروس كواتر الليفر أتلك acting like a buffer or a bank for glucose كواتر بهوي الليفر هو يتوك جلوكوز ريت أبيت فين الجاكت فين أبينا ما قاعد تدوك مش هاي Look to this diagram or pictures. Show you how glycogen break down in the muscle and the liver. Goku bombas, and she can show differences are important. Liver can release glucose into the blood, while the muscle cannot release glucose into the blood. This steps of degradation of glycogen changed to glucose 1-phosphate, and glucose 1-phosphate changed to glucose 6-phosphate in the liver, and glucose 6-phosphatase found in the liver, while glucose 6-phosphatase not found in the muscle. That's why muscle cannot release glucose into the blood. This diagram shows you most of the metabolic process inside the human being are hormonally regulated. Hormonally regulated. Hormones play an important role in the metabolism. How? By activation. Look to this diagram. What is the title? Hormonal regulation of glycogen degradation. With the hormone plays an important role in the metabolism, driving most of the reaction inside the body. Example. ليانج ايجا لتمني ويا توزي جولتك توزي مناطق هالشيء خوي زور تأثير تيناك زور نابي بأوبيس أو خواردني كأخوي نابي بفات أبي بمصل يا أبي ببالا بلام كدو لدويتش صاليوا أبي خواردن خوي هي وخواردني كدوي خوي لجاتي بي بمصل أبي بفات how لبروي هرمونال there is imbalance in the hormone secretion. Hormonal and how kegel. لأيجي إيواء يشد يخوار بخوي بمضبوطي ميتابوليزم لمنا أخوار هاتشي أخوار زود كشيني بلام لقورت على برو هرمونالي يعني هرمونال بالانسينيا أخوار ديك إخوة أبي بفهم. This diagram show you hormonal regulation of glycogen degradation. You see insulin. This plus sign means activation of glycogen synthase. So insulin aid in the formation of glycogen. Aid in the formation of glycogen. While the other hormones, like glucagon, epinephrines, AMP, will stimulate glycogen phosphorylase. In case of stress, there's a hormone called glucagon and epinephrine. These are hormones of fight and flight. Stimulate degradation of glycogen. While the other hormones, insulins, stimulate formation of glycogen. This is one of the examples of hormonal regulation of metabolic process. كواتلم diagram what she developed. Insulin stimulate glycogen synthase and the branching enzyme glucose marker, while glucagon and AMP epinephrines stimulate glycogen phosphorylase and debranching enzyme. كواتا كاتي stress كاتو stress هي شرك ايام برسيتا بيسبب جلوكوزا activation او enzyme انا. How تينا deep dip. لما حضرتك يشوتنا باس مانكر phosphorylation and dephosphorylation will activate some enzymes like this one, glucagon. Glucagon will phosphorylate an enzyme called glycogen phosphorylase, which become active, while glycogen synthase became less active. Counter phosphorylation changed the enzyme activity. Phosphorylation of glycogen phosphorylase became active and phosphorylation of glycogen synthase became less active. While dephosphorylation of this enzyme, oxyamine, became less active. Insulin causes what? Stimulate protein phosphatase. Remove the phosphate. These two pictures show you hormonal regulation. كمان أرزاني glucagon, insulin, AMP, epinephrine شيكا. لستيبي دوما, the diagram دوما. Phetalate phosphorylation and dephosphorylation of the enzyme make them active or non-active. هموش علاقه با چی وای glycogenolysis. لکاتی بسیتیا چون گلوکوز بسیته و بهم هورمونانو. By which mechanism? By phosphorylation and dephosphorylation of the enzymes. What about the glycogen storage disease? Harrow point of view, we have different types of glycogen storage disease, in which there is difficulty in the breakdown of the glycogen to glucose. This is a problem, yes, because this leads to hypoglycemia, and hypoglycemia is a very dangerous condition. Anyone can define hypoglycemia or blood glucose level in case of hypoglycemia? At which level glucose is called hypoglycemia? Below 40. Below 40 is hypoglycemia. دائما normal range وأوشانا مهمة بلان أو مهمة. Below 40 is hypoglycemia. Hypoglycemia, if untreated, it will lead to brain damage and death. If there is accumulation of glycogen in the tissue, accumulation has substance in excess, in any tissue, it will lead to damage to this tissue, like damage to the liver. Agar tissue glycogen زيادة أو better هوشي hepatomegaly or liver enlargement, وهو galactosemia. And poor exercise tolerance. سيرك منالك فكاني هم يتواني يعني كان والله أو يكتوز رابطة هناك بياك بعذاب يتو. What about lipid? أو باسي carbohydrate. بوتوزك ما بو باسكتن. Just a small idea about glycogen and just small idea about the lipid. What are the differences between lipid and glucose? Accumulation of glycogen in milligrams. هيك خلاوة بيت. الله نقول جيتو زور زلام نقرأ glycogen. نعم. يلا نقرأ لفات. Accumulation of lipid in the form of kilograms. The kilogram شوريكو بيتو. They are hydrophobic. They are structural, found in the cell membrane. They are important for the hormone productions. They are called lipoproteins in the blood. They cannot be transported in the blood alone. They are combined protein with protein called lipoprotein. Lipid plus protein called lipoproteins. Stored lipid with manchia, triglyceride. These are structured just for simple memorization. You see, we have different types of fat in the blood. Fatty acid we have triacylglycerol. We call it cyanide cyanide سيرك إلى شيء glycerol by one and 3 fatty acid. Fatty acid triglyceride. We call it cyanide, we call it triglyceride. Phospholipid are complex lipid. Lipid plus phosphate. And cholesterol are good or bad? Basha? Cholesterol is good for human, while oxidized cholesterol is bad. Cholesterol is important for the formation of cell membrane, hormones, and they are very important for the formation of bile. And also we have another type of lipid, carbohydrate plus lipid called glycolipid. Steroid. With cholesterol steroid or sex hormones, mostly composed of steroids. See the mass of this compound. Someone 70 kilograms, approximately they have 15 kilograms of fat, 0.4 glycogen. What the glycogen zor kam here, and muscle proteins. In case of obesity, 80%. So the body composition is very important. Body composition is very important. كواتاتو دوزا لما بيني يدي كان healthy يدي كان healthy عين كيش يعني عين كيش يدي كان فاتي زورا body fat percent is very important. Body fat percent يعني أبدو بزاني body fat percent يخول شنا. الإنترنت ليبا بنوسا pictures of body fat percent. دوزا لما كان شنا حتى low body fat percent بي عمري دريشتري بيتو يعطي شيء كم زيادة بيت. تافات كم بيت. تافات الزور بيت. cholesterol برز بيتو directly related to the health and long living. كواتا قد نلا ينزل ما قلوا والله يسقاني استولش يواني الدنيا. يستانس وريني أو قلوة خرافة أو ضعيفة باش بلام نوع تريشة من باسي body composition كم body composition شيء مش كتير معناه بزوري لا إيمة زوري أمشتا بيوتي skinny fat شيء ضعيف شبه شوري زورا. It is important to know how you measure your body fat composition. The major type of lipid are triglycerides. كوات حكيك أخوي دو triglyceride تشوفي بزوري. لكرا لشايا. How they are metabolized? They are hydrolyzed by the pancreatic lipase. Pancreatic lipase. That's to say, pancreas is an important organ for the metabolism of lipid. If someone has disease in the pancreas, what happens? Aggregate pancreatic lipase in area.
Look at the metabolism of fatty stool.
Fatty stool. How you know you have a fatty stool? شلون أعرف fatty stool هي؟ أنا جاي كنت جايبي وش بنوع.
جايبي من لكمان.
It is the metabolism of fat. Bile acid play an important role in the metabolism of fat. Bile acid. كواتك يجي عمليات استراحة وهي bile acid كما توشي استياتوريابه. توشي استياتوريابه. كواتك سلغي فات يعني. Woodman triglyceride is composed of glycerol and 3 fatty acids. Glycerol and 3 fatty acids. Glycerol لكي وهاتوا؟ Glycerol كوي لكي وهاتوا؟ لجلوكوز وهاتوا. لجلوكوز. زور كسيه بولا بالله دكتور من شبريكم هذا نابض. تبي شيء بيبلي؟ شيريني ما. شيريني نكتشوي. Glycerol is a glucose derived or arrived from the glucose. You see glycerol changed to glycerol phosphate by the action of glycerol kinase. Kinase put phosphate. And finally, this glycerol phosphate changed to triacyl glycerol. Glycerol is a backbone of triglyceride. كواتك هيك triglyceride يبرزت شو يجي يصل شيء بيلا أبي بيبلي glucose يا شيريني كم ترى شيريني كار كم ترى. What about the fatty acid? Fatty acid, we have different types of fatty acid, saturated and unsaturated fatty acid. Unsaturated means they have double bond. حتى double bond زوربة لفاتي أسيدا كين زياكا solubility زياكا يعني رونكا أبي الشل أبيته ولا جاكتيويكا hydrogenated يعني رونيتون حتى double bond ديكي أبي الشل أبيته which is important for the fluidity of the cell membrane كواتا حتى double bond زوربة cell membrane رقنابه كcell membrane رقبو معناه أولا كتو توشي أثيروسكليروسا توشي نقوشي بكواتا أوميغا 3 فيش فاتي أسيد أوانك أي خوي أوميغا 3 تي تها دبل بوند دي تها أوميغا 3 يعني شي دبل بوند لرقم سي لرقم سي جاي وادو أوميغا 3 كتو يمسوا سي شش نوع كتير يمسوا سي شش نو لأن لم بوزيشنانا دبل بوند human being can form only 3 and 6 double bond. No, not one, but they are obtained from the food. كواتا غير أوميغا 3 فريبو وكسر يا بو خود يا بو بس هيك إذا دبل بوند كاني لشوي كزورتر بو دبل بوند كاني بيكرا. Look to this diagram. Just write simple note. Simple notes. we have a process of transport of fatty acid from the cytoplasm to mitochondria. Fatty acid oxidation is important. One year? Important here fatty acid oxidation there. Agar to fatty jism, oxidation there. Very good, because it produces large amount of ATP and get rid of the excess fat. همو fatty jism أروات الدرو. حتى fatty can be باشترك. Oxidation of fatty acid is important. Fatty acid should be transported from the cytoplasm into the mitochondria. Why oxidation be? This man here, we have a transporter called carnitine palmitoyl transferase, which transport fatty acid from the cytoplasm to mitochondria. We have CAT1 and CAT2. Again, one man have if someone have CAT1 deficiency, if there is no CAT1, again, I'm going to
ناتجة السيتوبلازم ولا ميتوكوندريا وميتابوليزم
ناتجة mitochondria ولا cytoplasm. There is accumulation of fatty acid inside the cytoplasm, swelling of the cells, and damage of the cells. We have a fat burner here. Carnitine is used for burning fatty acid. يشوي ياريبكا يشوي عرضي يكمدو مواناته carnitine روجي دنكي اخوة would stimulate or enhance oxidation of fatty acid. أما ورقة CAT1 وريقليو وإم عملية زيابكا كواتا pointy أما تشيبو fatty acid oxidation occurred inside the mitochondria by the help of a transporter called carnitine or carnitine palmitoyl transferase 1. I got the baby membrane inside the matrix by CAT2. Hello, Basha. أو كارت شون لبيتها ناشيتوا شون الله كار إيشي تيا consumption بنزين يعني قاسنا وكوتي كارنيتين يكسري كارنيتين بوا أزاني oxidation fatty acid transport يكاتو سايت ميتوكوندريا باشا شو لبيتها ناشيتوا لا قبل بيتها ناشيتوا inside the mitochondrial matrix there is oxidation of fatty acid. We have different types of fatty acid. According to the length, there are several reactions occur inside the mitochondrial matrix in which removal of 2 carbons of the fatty acid. I'm cyclic 2 carbon درجة. I'm هشدة. Fatty acid هي هشدة. بده oxidation يبصرها. 2 carbon اللي أقوى. And production of FAD and NADH. I'm say ATP. I'm do ATP. I'm cyclic. كواتا حتى oxidation زور حتى كربون كان نمبر زور تبي cyclic زور تبي ATP زور تبي كواتا أواني كبرنامج كيتو كان على إما energy زور مانيا تبي fatty acid oxidation هو زيادة. There is no glucose. There is oxidation of fatty acid and production of large amount of ATP. Rasha, ama oxidation bu. This diagram shows the precursor of acetyl-CoA, which is the backbone or precursor for several substances like triglyceride, phospholipid, ketone bodies, steroid hormones. acetyl-CoA هنا نفسها acetyl-CoA بكاريا بودروس كدني steroid hormone triglyceride phospholipid ketone bodies. Ketone body chia. In case of severe starvation, يعني بس فار بكارين أو إنسان زايد كيتو بكارين رجيم ketone body بكارين لجاتي glucose which produce large amount of ATP. Look to this diagram and write short notes only, short notes. Fatty acid oxidation and fatty acid synthesis. They are different in the location. Fatty acid oxidation occurs inside the mitochondria. كواتا اگری که مایتوکندریا زور به اکسیدیشن زوره به وانیه کواتا فاتشی زور اکسیدیشن به چی مایتوکندریا مازیاقی نیما به چی انسان مایتوکندریا خوی زیاده
ستريس والتمرين
کواتا استرس نامنه هر هست
انا بالتمرين بالتمرين كده بس
increase muscle mass حتى muscle زور به مایتوکندریا زیاده او کواتا فاتي اسيد اوكسيديشن هي محصولکی زوره خواردنی شخوا بلام مایتوکندریای زوره البته هوی اکسیدیشن بلام بس اینو باسی چیه این fatty acid formation fatty acid synthesis occurs inside the cytoplasm لچی دروسه بیاد عادة كدوش كيف دروسه مثلا كيف دروسه كيف الله يلسه هيكو لأردو لشيرو لنازانم شي ألا ما دروسه بيه ببزاني fatty acid إيش لچي ودروسوا acetyl CoA وتمان لزور reaction كان هو acetyl CoA there NADPH which is the reducing substance CO2 available everywhere كهم شوية CO2 هي لقل ATP أمانة عادة لشو هي inside mitochondria طبعا لميتوكوندريا أبي بيت دروا بالسيتوبلازم This man beta hetero, la regici hetero, acetyl CoA change to straight, and also we have a shuttle, مقطع shuttle شيء transporter يعني transporter. سعري كيكتشوكا وتري shuttle. لجاتي تاكسي لبيني تاكسي وقاصة شيء كيبيو shuttle لفرنسا. أيوزيت وا. Straight shuttle. Difference legal evitirana chibu carnitine shuttle. Straight shuttle to transport straight from inside the mitochondria to the cytoplasm. In the cytoplasm, we have an important enzyme called acetyl-CoA carboxylase, which is the first step in the formation of fatty acid. The most important enzyme, which is called top gun, يعني سلاح بعشرة 20 سلاح يعني top gun acetyl-CoA carboxylase, which is also hormonally regulated. Hormonal regulation is very important. وكبون باسكت. هي الله هالشيء كخوان يسوتين بخيال أنا هاي شوية الله. وكي يسوتين hormonal regulation بعشرة. How? Glucagon, which is a stress or fight and flight hormone, inhibit this enzyme. كواتا قاليك stress يبين معدل راكدنو معدل خوي ياكدنو أماني هبه fatty acid دروس نابل ضعيفة بيتهم. Insulin stimulate the formation of fat. Insulin is an anabolic hormone. And finally, formation of fatty acid. هالشيء زائد كواتا stimulation acetyl-CoA carboxylase glucagon, insulin, citrate, والمانونيل-CoA inhibit acetyl-CoA carboxylase. fatty acid will form inside the liver. Finally, exit liver as a form of triglyceride or VLDL. أبيبتشيت الدرو بوبلا fatty acid. We have a drug that prevents excretion of fatty acid from liver to outside to the blood. شيء نايله fatty acid بيت الدرو لليفر. درجة الكحول. هذا أوانيك الكحوليزم أوانيك هذول الكحول لكونه شيء يعني fatty liver. One of the causes of fatty liver is alcoholism, which prevents excretion of fatty acid from liver to the blood. Lemla show the carbohydrate man. If someone eating large amount of carbohydrate or sugar, you see glucose change to pyruvate to the inside the mitochondria. This is stimulated by insulin. Insulin سيرجع همو إشكا أمريكا. Insulin glucose أبايكا بفاتي acid excretion بين liver. So أنت أكشف glucose source one. Glucose أكا أبي بفاتي acid. Fatty acid inside the endoplasmic reticulum, we have desaturation and elongation of this fatty acid. January fatty acid can do dehydration. Fatty acid formation and fatty acid degradation, they are two different entities. One occur in the cytoplasm, one occur inside the mitochondria. Location are different, and they are not the reversible. يعني هاد كان يعني عكس يكنين بلاي أو دروس بتوش أو أقربه. هاد كان لدوش يعني جوازي. Fatty acid formation is anabolic. Degradation is catabolic process. سؤالي fatty acid oxidation goes into nutrition. ما important زيادة يصير.
دقيقة ما تاخذ أنا داخل بيت البنت. دقيقة داخل. Okay, today is كان ما هو جاي بيضان بيت البنت. أول حاجة بقول قلت أنا محاضرة بيضان بيت. إيش اللي بسكرتني So this will be the last lecture regarding the carbohydrate metabolism, and it deals with the electron transport chain and oxidative phosphorylation. Actually, it's not specific for carbohydrate, it belongs to every other component regarding amino acids, also fatty acid and carbohydrate will end up in this pathway that's called electron transport chain and oxidative phosphorylation. They are a combination of two pathways. Later on the figure, I will tell you what's the meaning of oxidation and phosphorylation pathway. So, if you remember, I showed you this figure on first lecture, and I told you I will tell you the detail later on. We said that the major source of the energy in our body is carbohydrate, protein, and fat. We talked about this part. We talked about carbohydrate. We talked about how the carbohydrate has been digested and converted to glucose to be absorbed, and how glucose converts to pyruvate in glycolysis. And then pyruvate was seen in an enzyme, PDH, converting to acetyl-CoA. And we talked about TCA cycle as well, how it will give off to CO2 and many, around 12 ATP per cycle. They will give off in a form of 2 NADH, 3 NADH and FADH2, and in form of GTP and ATP. Then these NADH, I told you that they have 3 ATP in each molecule of NADH and around 2 ATP-ish in an FADH2 molecule. So they will go to this cycle that we'll talk about it today. It's called electron transport chain to form ATP to be used for your function. So this is how we extract energy from these two molecules, that's called NADH and FADH2, in this cycle that's called electron transport chain to convert it to ATP to be used by our organs for various functions, of course. So this is what we talked about in carbohydrate metabolism. Today will be the last part regarding the fourth step. It is electron transport chain. So what is oxidative phosphorylation or electron transport chain? Is a stage 4 of metabolism, as I showed you on the lecture. Is the last stage on the figure, sorry. And it's a complete oxidation of glucose that yield only 2 ATP and 2 GTP. In the glycolysis, if you remember, we have only 2 ATP, and also in the TCA cycle, we have only 2 GTP. The other energy reside in a form of NADH and FADH2. So we have to take a benefit or use these NADH and FADH2 to generate ATP for our function. Otherwise, we have only 2 ATP and 2 GTP. Where has all this energy gone? As I told you, it resides within NADH and FADH2. So we have to convert them to ATP to be used. So the energy is still carried by a carrier, mainly NADH, طبعا زوربين cycle كان NADH second with a minor degree FADH2. So they will combine with the oxygen. I will show you on the figure now, it may be vague how they combine with the oxygen. They will give their electron to oxygen, generate water, and they will be, in this process, when they give their electron to the oxygen and generate water, they will release energy in the form of ATP. Again, also FADH2, they give their electron to oxygen, generate water, and the energy also released in the form of ATP. This will be discussed in detail in the next figure. By the way, around half of the energy in the electron transport chain will be released or will be used to generate ATP. You may ask where the other half has been gone. NADH يكسانا بس ATP. بلان خوي للراستيا إنرجي كيزور لو زيادة دروسك على ATP. بلان بيوي إنرجي NADH بكار هندرت الإلكترون ترانسبورت تشين ودرس كني ATP. نيوا كيتري تشيليه تقريبا بعض نيوا 50%. Yes, generate heat. Why? Why do you need? Yes, very good. So the other half will be used will be released as heat. Is it wasted heat? No, it's not wasted heat. It will be used to maintain our body temperature, okay, with a normal range. So how they will be oxidized? It's carried out in a pathway called oxidative phosphorylation. Oxidative phosphorylation represents two processes. Oxidation of NADH and FADH2 electron transport chain. Essa qsana hamwi nakhosho complex بلاتان. بلان كتشينا سفي جركا بوم باسكت نيك بيك وصار ركينو دواتر كأشنو سرتوكا كان أساتر أبد. So oxidation of NADH and FADH2 in electron transport chain. ليرا electron كاين سر NADH و FADH2 أجوزريتا electron transport chain. And then there will be phosphorylation of ADP to produce ATP. How we will discuss it. So by definition, as we said, is a final step in every metabolism. In fact, amino acid, carbohydrate metabolism, in aerobic cells. Of course, the cell should have mitochondria in order for electron transport chain to happen, by which electron derives from various substances and liberated to oxygen, which is reduced to water. So oxygen finally converts to water, and the energy is used for phosphorylation of ADP to ATP. The electron from NADH and from FADH2 will be put on oxygen to generate water, and that energy will be used to generate ATP out of ADP. What's the location of electron transport chain? Is it in the inner mitochondrial membrane. طبعا مجموعة location ما بس كده cytoplasm the mitochondria, but this is happening exactly in inner mitochondrial membrane. This is just a little overview or a summary of how this is happening. As we know that most of the pathways, the pathway كان كخدمة لنا glycolysis لوانه TCA cycle PDH چی دروست دەکەن زۆربیان NADH دروست دەکەن هەنێکی لە TCA cycle FADH دروست دەکات بە شێوەیەکی minor بە شێوەیەکی سەرەکی major substance کە دروست دەبێت کۆمان دروست دەبێت چیە NADH باشە ئەم NADH چی دەکات ئەلکترۆنەکە ئەخاتە ناو electron transport chain بە چی بە NAD خۆی اوكي كالكترون ليرا فلو اكا بم تشينيا كاذا باسيك بيغاتو دا چند كومبلكسه كليرا فلو اكا الكترون انرجي ليجي ابيتو اوكي ام انرجي بكار هنري بودروس كردني ATP ل ADP اي الكترونك خوي تشي دير بلو انرجي الكترون اچ دسو اكسجينوم ببا ووتر او اكسجيني كديليفر بلان هيموجلوبين that's why we breathe oxygen in the first place the better oxygen اما breathing yanking بوشي لبروي ام الكترونان يبقيتو ووتر ما بودروس اوكي انرجي اللي جابته بوشي بكاريا نيوي بودروس كدي ATP نيو كيتريبو heat to maintain your body temperature within a normal range okay this is the summary about the electron transport chain and how it works تيجيشتو لمبشا لمسمريه okay so this is the actual pathway regarding the electron transport chain and first of all, to orient ourselves on the right side, we have the mitochondrial matrix. In the middle, that actual pathway happens, is inner membrane of the mitochondria. And then we have on the left side, we have cytoplasmic side, okay? So we know that the most of the NADH have been generated in the mitochondrial matrix from the TCA cycle. Or we can say from other pathways like fatty acid oxidations or protein metabolism that you will get it in other lecture. But regarding the carbohydrate, most of the pathway regarding carbohydrate is TCA cycle and PDH pathway, generate NADH. So once the NADH has been generated, يجب مثلا pathway كذا بسرعة ثانية كم mitochondria شن membrane هي؟ أي بوشي باسي outer membrane ما نكجوا؟ يجي كذا زاني لصوبه
لا
Yes,
transport len the outer asantra
It is a highly porous membrane. We don't need to worry about the outer membrane, so we have just an obstacle regarding the inner membrane. فهاتنا مشكلة كمان ليرة the inner membrane. The outer membrane highly porous as well. Molecules freely go in and out, so we will not talk about the outer membrane. So we just mention here the inner membrane, okay? So once we have generation of NADH from various pathways, as we said, on TCA cycle, for example, a PDH pathway, they will give their electron first to the first complex in the electron transport chain that's called NADH dehydrogenase or called complex 1. Electron ki khoya bam complex so khoya bada chi be NAD u jay gidir bakar henjito. Okay? So later NADH kala pathway kan drusbu electron ki yaba complex 1 which is called NADH dehydrogenase and then convert back to NAD to be reused by the pathways again. So this complex will take the electron and go to the complex 2. Before going to that, okay, it go to the NADH, give the electron through this chain, that's called electron transport chain, to coenzyme Q or called a ubiquinone, or sometimes you may hear it's called Q or CoQ10. So it's many names for this protein that's called coenzyme Q or ubiquinone, take the electron mainly from the NADH dehydrogenase or complex 1. يجري بردان يمكن أيام كومبلكسي سيليرا سي bypass reaction مانيا. bypass reaction يعني شيء يعني bypassي كومبلكسي يكم مكان. أوكي؟ شلون؟ بونمونا أجر بيرتعمل TCA cycle شلونك ما نهبوك FADH تدير بس. أو بعتت من إنزيم كم مهمنية. بلام ليرا نامي succinate dehydrogenase بيوتر complex 2. Okay, so am enzyme چی درست کن؟ FADH درست کن. راستم خو الکترون کن ایاب چی با coenzyme Q, bypassing the complex 1. Also, this is present in the TCA cycle. Also, in beta oxidation of the fatty acid, we have an important enzyme that's called fatty acyl-CoA dehydrogenase. Generate FADH to give it to coenzyme Q again. Also, in the cytoplasmic side of the cell, we have glycerol phosphate shuttle. Generate FADH2 and give the FADH again to the coenzyme Q. طبعا FADH مقارنة ب NADH بالركيز زوزو كمتر للسيل كان. So they are minor in number compared to the NADH, but they are bypassing complex 1 directly to coenzyme Q or ubiquinone. بيجوا كأم رسم بجبلي طبعا ليرة drug كمان هي درجة PO2 barbiturate يعني تقريبا anti depressant يعني hypnotic drug يعني بعامل عليها بوز and has many use. Again, overdose به شيء أكا barbiturate inhibit NADH dehydrogenase complex 1 يعني retinol which is an insecticide if cause toxicity it will inhibit complex 1. فسارة كلها أويا أما هيج ATP دروساكين كاتك barbiturate inhibit NADH كات NADH dehydrogenase كات complex 1 كات ATP دروساكين balance how
In glycolysis and in the TCA
yes, they are minor ATP amount. Regarding the electron transport chain, do we make any ATP in electron transport chain when there is barbiturate around? Yes? Yes, we do make ATP, but a very little amount. Because as I said, these three pathways are bypassing complex 1. So even if you are inhibiting the complex 1, you are still making ATP, but a very small amount of ATP, because most of the cycle generate NADH. So it's not a good life for the cell or for the patient, because you are obstructing the major generator of the ATP, which is NADH. But still, we do make a little amount of ATP in the presence of FADH2 in a minor bypass reactions. Okay? تقرأ واضحة جدا؟ أوكي. دواء بك إلكترون كان يلعب كوإنزيم كيوبيكوينون أشيبو كيو أشيتا complex 3. Here's not going now in the back. Had a complex 3. That's it. Okay. So they will give electron to complex 3, then from complex 3 to cytochrome C, which is present on the cytoplasmic aspect of the cell, and then electron will go flow to the complex 4. أولا now complex 4 مهمة زي ديناميك المجموعات ناري هي هيش كريسناك أو حبيب زانن. Just know that they have an Fe, what they have an iron in a +3 state. Complex يتوار iron يتعالى ل +3 state. إستفادي شيء. كانت الإلكترون كان flow أكن بناو أم شينا. الإلكترون كان أشد السر iron ل +3 state. إكابة شيء إكابة +2 state. Okay? بوشي باش. أم +2 state يت لا أكسجين كوا إلكترونيكي خياطة بأكسجين أجورته ب+3 state. Okay? إلكترونيكي تريت أشد السر complex يتوار iron +3 كاتو بشي +2 state. +2 كإلكترونيكية بأكسجين أجورته +3 state. يبردام أم حركة هي +3 +2 +2 +3. Okay? The aim is to transfer electron from the complex 4, which is a low energy electron by the way, to transfer the electron from complex 4 through the iron to the oxygen that we breathe and generate water. Okay? Complex 4 again, it has iron in a +3 state. Take up the low energy electron, convert to Fe +2 state. Okay? So what you look there at Shashtania? What you want there? Alright. تعطى إجورة بشي إجورة Fe +3 إجورة Fe +2 Fe +2 جت لا أكسجين إلكترونيكي خوياتي إجورته Fe +3 إجورته complex 1 أم يشبه دوام. Okay? وين اللي رأم مزور تأكيدي لي أكينه هلا بالقوي there is a killer here. It's called a cyanide and carbon monoxide. That's why we say cyanide is a lethal gas or it kills people. Why? لبرو ليه cyanide باين شركة باين Fe +3 أكا. كباين Fe +3 كله كومبلكسي شوارع جاري كتير ناتوان إلكترون جيزتوه أكسجين. Okay? كاتل ليه استكبر ليه pathway كيتو أو step ناتواني ATP درسبي so the person will die from cyanide poisoning. كاتل بوشي لبرو cyanide كليرة كاتل بوشي بس Fe +3 Fe +2 ما كده لبرو ليه أجل سيكين cyanide معناه لقى carbon monoxide. Cyanide يباين تشيكا Fe +3 in a complex 4. كباين ديك تشيكا you have a big X on a complex 4. So no more electron transfer to the oxygen. So everything will be blocked here. No more ATP production and you will die from cyanide poisoning. Okay? Then هذا بالأمريكا cyanide poisoning lethal fatal. بعكس أول and last شيء بتفاصيل carbon monoxide. Carbon monoxide باين تشيكا Fe +2 state. كاتك Fe +3 كها أكسجينكي ورقل تيجي بيا بأكسجينك carbon monoxide يا باين ديكا. نال أكسجينك كاربون بريبو إلكترونك بريبو أكسجينك. كاتك بحماش يو carbon monoxide poisoning هەر ئیثڵە هەر ئەبێتە هۆی مردنی کەسە باشە چکە ناهێڵە پرۆسەی گواستنەوەی ئەلکترۆنەکە بۆ ئۆکسیجین بەردەوام بێت تێگەیشتن بە بەشە Yes. Kamian, cyanide of cyanide. Okay, cyanide, which we said will bind to Fe in a +3 state. Fe +3 is a complex trial. Kawata again, mint blocky pathway, was no electron can bo oxygen. Kawata, there will be big X on this complex fork. No more flowing of the oxygen to the for the flowing of electron to the oxygen. Kawata kalera pathway come was معناها هموشتر ليرا بلوك بيت فبتجي تشتر فلوي أكسجين إلكترون نابت كواتا لن pathway ناتوني بردام بيتو ATP دروز كان كواتا ATP دروز نكد بيشتكا تشي ليا they will die because of the cyanide poisoning. عشان بو أكسجين أم إلكتروناني كبيرة كالإلكترون ترانسبورت شينا هم يجي تستر أكسجين كي غورة بوت. We said this is a low energy electron, okay? Low energy. باشا بالسيارة كلها أوي أو الإلكترون energy كي تشيلي هات. تشيلي هات. بس ID هي لباري أو بيجي تتسر ATP تتشوني كتير. أما بين الليرة أما تشيل. بروتون. كواتا أجر تصوري أو كان NADH NAD واتوقعي كان H كاتولا electron and proton, okay? الكترونك لو انرجي الكتروني يد ايام اكسجين انرجي كنا وسطرتي كيو شو تناو بروتون توقعي كم شلون نظام الله ما هو يقول لك كيف عن ايش هذا مين كان هي ماجيك كده sorry look at that اگر imagination كان شيء زور آسان بقوليه سيكل سيليكا باوكي بسمع NADH لبعضوية كأقوربة شي أقوربة NAD وإيولش لنا تفاصيل الكيمياء وشون أشتغلنا وأعرف شو براسي I don't encourage that because they're medical students actually you are so MH أكيد بدوش electron and proton okay electron كبر شو أنا electron transport chain شو finally oxygen while أما low energy electron وشي تبي energy كي هم اللي شو أقول بدرو now proton كأ أسبي أوتين proton okay تيجيش now إيش شركة we have NADH will convert to NAD. The H part of NADH will split into proton and electron, okay? So the electron part is low energy electron now going to the oxygen to generate water. Now the energy is reside in what's called a proton, okay? So this side, it's called a proton. How am energy أنا قد يقتي رسمي سلوتريش بي سيريكا شوني هو ليرا كإلكترونك هاتوا لم لا تشي درجوا لم بالمقابل بروتونك كان هموي through the inner membrane go to the cytoplasmic side. Now generate what's called a gradient. ليرا تشي دروسا كان gradient دروسا كان يعني شون gradient يعني للايك بشكل زور بروتون مان هي للايك هيش مان هي. كواتا now these protons they want to come back to the mitochondrial side in order to balance the equilibrium. إذا أم بروتوناني كهمو إنرجي كتير أنا بقرنه بو mitochondrial side. وش يشكل لي gradient يدرس بو يعني جوازي يدرس بو لا concentration كيا. كواتا أنا هم يار بقرنه بو mitochondrial side. شو نقرنه؟ Through this bleb that's called F0 F1 compartment. لري أن compartment ده كأنه ترى F0 F1 compartment. Okay? كواتا إستا بروتون كان كإنرجي كان هودرتوها همويا لري F0 F1 compartment ده أقرنه. أم كنالايان أم component تشيتيايا إنزيم كتابة ATP synthase. ميوي إنرجي بروتون كان ورقريبو خويو تشي دروسكات ATP دروسكا from ADP and inorganic phosphate. Okay? تايرة واضحة؟ تيجيشتون؟ كسي برسالي هبه اوكي كواتا ليرا شي ما مود رزبو انرجي كمان هاتو تناو proton gradient ما مود رزبو ويبقى to mitochondrial side we require this bleb that's called F0F1 compartment. They have ATP synthase enzyme يعني they will synthesize ATP. Okay, this ATP synthase will use half of the energy of this proton to generate ATP from ATP. So this is how the ATP is generated from the flowing of the electron and proton gradient. So we said half of the energy is used for generation of the ATP and the other half will use for release as heat to maintain our body temperature. Yes? But so, طبعا كاتك ATP قد رزبو ATP معناه بيوتك translocase. It will translocate the ATP back to the cytoplasmic side to be reused again. كATP زور أبيلرا يتوناو matrix side وأويك يبقى تولى inorganic phosphate ATP بيدرسها. كاتا ATP يتدرو بكار هذا ATP يتناو ATP يتدرو ATP يتناو بوي بكار بينه بودرس كبني ATP زيادة بوصل كيتو. أوكي؟ بوي لرا ATP برespiratory control داني. بوشيش كاتا level ATP زور تربيه. كاتا معناه كويس هم cycle بس ريعتي إيشكا كويس هم بوي ATP زور تربيه. That's why it's called a respiratory control. طبعا ليرا دروجك معناها بيوتر oligomycin inhibiting channel أكا بيوتر F0F1 compartment. كاتا channel block بوشي أبد ATP دروزنا بجاري بيتر life patient أكا كرة خطر واحتمال pass بوي oligomycin toxicity. If there is any question till now, oligomycin is a toxic macrolide. It will inhibit the F0F1 compartment, so it prevents ATP synthesis, so you will be die because of that inhibition. Yes. Alright. طبعا مجموعة أشغال هي ليرا بيان أو تري uncouplers. Okay. Uncouplers شيا بيشوا بوتان باس كم Oxidative Phosphorylation بس بلد. Transfer ATP back to the cytoplasmic side to be reused by the organ. Okay? So it's translocate the ATP. Okay. Okay. طبعا مجموعة أماني بيشترك بأصلاً cyanide, carbon monoxide, oligomycin. They are called inhibitors of electron transport chains. While now we have what's called uncouplers of electron transport chain. What's the meaning of uncoupler? They will uncouple oxidative part from the phosphorylation part. So meaning the electrons are still flowing, but there will be no generation of the ATP. So this is the meaning of uncoupler. They will uncouple the oxidative part from the phosphorylation part. Electrons are still flowing with no generation of ATP. Okay? Nimone uncoupler kan kim 2,2,4-dinitrophenol, chia, is a weak base that allow H back to the matrix. طبعا 2,2,4-dinitrophenol أو كشي إيشكا وكو F0F1 compartment إيشكا. كاتك أما هبل لوناوا ناهل لك بروتون كان بينه F0F1 compartment خوي إباتو ناو matrix side. كاتل ليرا ATP دروزنا بلام still the electron are flowing. فتري لما بيش أم دراجة أموكو دراجة 2,2,4-dinitrophenol بكارها هاوكو anti-obesity medication يعني بكارة how about treatment obesity. طبعا زور سو أم research يعني بلين أم trialan وستان لسه patient تكا. بوشي وستانيان anyone has any idea شكوز ما 2,2,4-dinitrophenol شيء كات إلكترون كان والي أكاك flow كات بيرا bypass the F0F1 compartment. كات إلكترون بردان flow أكاك بلان هيج ATP دروز نابت. بوشي برأيي أو أم trialan وستان وكوكو anti-obesity بكار بينره. شي هبل؟
Fat accumulation.
نخير.
Energy درست نه.
Sorry?
بهزي.
How you explain it?
ATP mania.
No. فين قشتن؟ سينيكا. 2,2,4-dinitrophenol شيكا. إيشي F0F1 compartment لكا. بلان جكلويكا. ATP synthase enzyme هي تا ATP درسكا. كواته همو بروتون كان باتوا دي matrix side يعني دي mitochondrial side ببدرس كمين ATP. يستعمل كارها بو ديسك بو انتايوستي ميديكيشن بوش بكارها بو انتايوستي ميديكيشن دبل يقطو اي تي بي درزنا يدن شي بزلتو اي دي بي بزلتو اي تي بي برزبو بردام فلوي اوكسيد الكترون بردام بكاتا تو بيرلي فاتس زورتي بكاتا مشتي كي باشا بو اوبستي بو ان يكسايد افكتي هي كاجر دي قد من بيموتن بل افر دبل ويكا نخوشكوت معنی منی وی انرژی که بکارین ریبو ATP من الیرا اگر ATP دروز نکم 100% انرژی که بکارین ریبو چین will be liberated as heat که وقتا نخوشکت احتمال پاس به hyperthermia elevation of the body temperature لبرو يكسر وستانيان وام درجه تشيد بكار نها وقتها وستي ميديكيشن توقع كان شلون يعني اگر ب F0 F1 compartment انا رواد يكسر لغي 2 to 4 دايناتروفينول بروا 100% of the energy associated with a proton will be liberated as heat, so the patient will die from hyperthermia, okay? So they stop this trial and they no longer use this as anti-obesity medication. نخير هو اگر 2 to 4 دانتور ودرك بكار بينرد ببيع أروات خوي half ATP thin will take the half of the energy to generate ATP. The other half will be, yes, of course, you have to have half of the energy in order the other half to keep your body warm within a normal temperature. I want to be hypothermia. Okay? Okay. كيس برسياري هي لسهيرة. طبعا Uncouplers كان high dose aspirin more than 150 milligram per kg will regarded as high dose. No need to know the dose exactly. So just know that the high dose aspirin may also may act as an uncoupler. You have another uncoupler. This is called as a natural uncoupler. This is called a thermogenin. طبعا دوايبو تنباسا كم لسر paragraph. لسر psychotic speciality هي تجيشتو. So what was the component of electron transport chain? Actually, I will not repeat most of them because I told you all on the figure. Just know that complex 1 NADH dehydrogenase will take electron from NADH to the coenzyme Q. Now we have complex 2 that will give FADH2. أنا خلال أبرنج كبو خوتانو لسر رسمة كان هلهم وياه. خوشتان لما ضبوا شيء سعيد يابكن رسمة كذا ضبط كان ولا سر رسمة كأتوان بيخنوا لسر في جرا كان implied كأ بيخناني اشتكي لتنا. We have complex 3. And we have also complex 4, as we said regarding the importance of the complex 4 is Fe in a +3 state, don't forget that. And we spoke about the proton gradient, 3 of the complexes, 1, 3, and 4, in addition to transferring electron, also act as a proton translocating complex. معناها proton أجازته both cytoplasmic site. They use the free energy to move protons from inside to outside of the inner mitochondrial membrane. أما هو بس كراوتمان كإلكترون flow أكاب electron transport chain لملا proton less matrix site أجزرته بو cytoplasmic site gradient رسكان. M gradient أماني بيجرينو بناوا لري شو أشتوا لري F0 F1 compartment. Okay. Now the proton translocating complexes transform the chemical bond energy of electron into electrochemical potential differences of proton called proton motive force. Put a big X on this paragraph. I don't need you to remember, okay? Now okay? شيء في الله مهمة جوجل باكل لما بس الامتحان شلون يتوشي مهمة من هولين زورتين وتان كنت كموا بقوي كتيب قال لما مفهومك نكله بيب كانوا بكرانه بشي امتحان ويكسرش بيتام شدو. So أجر تيب قال if you connect it to real life or if you understand it, it will be much easier. Okay? So what are the inhibitors of electron transport chain? Can anyone tell me what are the inhibitors? Yes, Abasman, yes, Keter. What are the inhibitors? Yes, what is the meaning of inhibitors? Chia can inhibitor again? Yeah, that inhibitor chia can? Inhibit what? Yes.
Inhibits the cycle.
Inhibit ATP synthesis, we can say. Yes, yes. So we have inhibitors and we have uncouplers. Inhibitors of electron transport chain, inhibitor of any step effectively inhibit the whole electron transport chain and oxidative phosphorylation. هلا بدايته باسي barbiturates ممكن كشون effectively inhibit ATP أكا باسي cyanide ممكن باسي carbon monoxide ممكن أما أنا inhibitor أكان. So they will inhibit ATP production in electron transport chain. That's why they are called inhibitors of the chain. So they result in decreased oxygen consumption. Why?
They very need oxygen to produce.
Why there will be decreased oxygen consumption?
Well, at the end of the cycle, we need oxygen for the.
We need less oxygen. Since we have inhibitors of the cycle, since we have inhibitors of electron moving through the cycle, so we need less oxygen. So there will be decreased oxygen consumption, increased intracellular NADH and NAD, or FADH to FAD ratio, because they will not be used by the cycle. The battery cycle كهم slow a bit فأعتبر شيء NADH و FADH تشيليه برز bit باشا. Decrease finally ATP production. So inhibitor كان شيء كان ATP production كما كانوا NADH و FADH2 زيادة على سلا كان شو كم تربك عليه cycle كان بعام slower. و finally oxygen consumption كان بيتو على battery electron flow in a slower mood compared to absence of the inhibitors. Important inhibitors, cyanide, carbon monoxide, oligomycin, barbiturate are all the inhibitors, but the most important one are these two. So about cyanide, it's a deadly poison because, as we said, it binds Fe+3 in a complex form, preventing electron transfer to oxygen. So electron flow will stop and there will be no ATP generation. That's why it's very lethal and deadly poison. So carbon monoxide bind to Fe in a +2 state in complex form. It also binds to hemoglobin, displacing the oxygen. Imagine hemoglobin Fe +2 state ويكبتواني oxygen delivered hemoglobin. So carbon monoxide also bind to hemoglobin. In addition, it's also bind to Fe+2 in complex form. شو هو Fe+2 in complex form? For Fe+3. Okay, very good. Okay, very good. Other inhibitors, as we said, are insecticides called retinol, oligomycin, which inhibit F0, F1 compartment, will decrease ATP synthesis. Also, we have another one called antimycin, it will inhibit complex 3, it's not important. What was uncouplers? What was uncouplers? Definition?
Oxidative part, phosphorylation.
Yeah, it will uncouple oxidative from phosphorylation part, meaning it will.
Electron transport continuous but not generate ATP.
Yes, electrons are still flowing with no generation of ATP. Okay? So uncouplers mean oxidative part will be separated from the phosphorylation part, mean electrons are still flowing in the presence of uncouplers, but there will be no generation of ATP. This is different from the inhibitor. Inhibitor همو process كهوا شكاته. بلام unccoupler am two part جياكاتو. Electrons are still flowing, but there will be no ATP generation. Examples: substances which uncouple electron transport with the phosphorylation of ATP. In the presence of uncouplers, what will happen? There will be decreased proton gradient. What should decrease the proton gradient a bit? What should? Yes, very good, because it has a way to returning back the proton back to the mitochondrial side. Okay, 2,2,4 dinitrophenol. كخوي proton كان أبعته وناو mitochondrial side. Decrease ATP synthesis, of course, because it will separate the two pathways. Increase oxygen consumption. Why? Because electrons are still flowing, so we still need oxygen for these electrons. And there will be increased oxidation of NADH and FADH2. So there will be decrease in amount of NADH and FADH2 intracellular. Yes. بۆچی لەبەرەوەی ئەنکەپلەر چی ئەکا خۆی ڕێگەیەک ئەدۆزێتەوە بۆی پرۆتۆنەکان بەرێتەوە بۆ مایتۆکۆندریال سایت یعنی bypassing F0 F1 compartment ئەکا. That's why there will be proton gradient will be decreased. Okay? لەبەرەوەی خۆی ئەیباتەوە بۆ mitochondrial site. So because the rate of electron transport chain is increasing, and electrons are still flowing with no ATP generation, so energy release 100% as heat. Examples, we have two types of uncouplers. We have chemical, some substance like, as we talked about, the 2,2,4-dinitrophenol, and increases the permeability of the inner mitochondrial membrane to proton, and electron flow continues. But ATP synthesis stops and energy dissipated 100% as heat. وكو باس مانكر كشون وكو anti-obesity medication بكارة هاتو شون دواتر stop يانكر لبروي كلبا hyperthermia or increase body temperature in a portion لبروي energy كلا صاصة وكو heat درجات لبروي there will be no ATP generation in the presence of uncoupler. We have another thing, it's called a physiological uncoupler. It's called a thermogenin. Thermogenin شيء thermogenin physiological uncoupler كان the brown adipose tissue هي the back of the neck of the newborn. أنا كبيبي يعني infant كتازة لداي كمان the back of neck of newborn شتي يعني بيوتر brown adipose tissue, okay? يعني brown fat cells, okay? أم brown fat cell أنا شيء تيا أم thermogenin تيا أم thermogenin إيش تيا it's called a physiological uncoupler. ريك عيني إيشي 2 to 4 داناتروفينول لكها بلا. برسالة كلها بوشي 2 to 4 داناتروفينول خرابة بوشي thermogenin باشا. لبروي thermogenin تنهان لو بشي back of the neck of the newborn هي بو قوي body temperature newborn كبيرة توبا normal, okay? تنها that's why it's called a physiological because it's present only in the back of the neck of the newborn in a brown adipose tissue and acts exactly same as 2 to 4 danatrophenol. Energy will be released 100% as heat, but only in this part to maintain the body temperature of the infant or newborn baby within the normal range. Okay? Okay? So, it's a natural uncoupling protein found in brown adipose tissue. It allows energy loss as heat to maintain basal body temperature around the and also present around the kidney, neck, breastplate, and scapula in newborn, but mostly it's present in the back of the neck of the newborn. All right. We have a situation called oxidative stress. So during oxidative phosphorylation, reactive oxygen species are produced by as a byproduct. بس ما نكون reactive oxygen species لا cycle بيشو لهكسوس monophosphorylation. طبعا الزربي pathway كان by accident they will be produced. Reactive oxygen species they need electron and they don't care where this electron is coming from. So they cause diseases, they cause problems to the cells and the patient or the person. So mitochondria produce superoxide radicals which may be converted to hydrogen peroxide. تقريبا نفسي أو processing كلا مثلا من باسكتين أتواني لما mitochondrial cell شادي باربته ولا هر area كأكو أكسجين مان هبيت chance يدرس بوني reactive oxygen species مان هي. Okay? Because there is electron are flowing with the oxygen. So some superoxide radical react with the nitric oxide to form these two, no needs. Just know these highly reactive agent can cause damage to the cell, particularly to the membrane. ريبوكو أوشويكا للred blood cell كان باس معك كشان بيتا hemolytic anemia كشان بيتا hemolysis of the red blood cell احتمال mitochondrial cell يدرس ببنه هو damage for the mitochondrial cell membrane. Okay? So fortunately, cells usually have an abundant supply of antioxidants. بس ما كل كيبو antioxidants يكزور زور باج ولا cell again. Yes. Bella? Chief? Reduce glutathione and where the electron are coming? Yes. Anyone who wants to participate? What's the most important antioxidant? Yes, NADPH. Yes, NADPH we said is electron donor. It gives to reduce glutathione, but red blood cell NADPH is a major source as an important antioxidant to get rid of these oxidative stresses. So fortunately, cells usually have an abundant supply of antioxidant which can react with reactive oxygen species to deactivate them in a various way. As we said, glutathione, NADPH, the balance between the reactive oxygen species production and antioxidant is important. And oxidative stresses now have an important role in wide variety of diseases. So في disease كان بهوي تشيو الرئاب ويزيادوني reactive oxygen species. So رئاب وباس مانكر G6 deficiency that causes hemolytic anemia and many other problems to the patient. Okay? And thank you very much.